Hello and welcome to our tutorial on metabolomics for quality control. Quality control is an essential process that ensures a product contains what it's supposed to contain and doesn't contain any dangerous or unwanted byproducts. Mass spectrometry can be a rapid and cost-effective way to perform quality control. Typically, this is done using targeted analysis, where compounds are identified and quantified using their mass and retention time. However, there are some drawbacks to this type of analysis. First, mass and retention time provide only 2 data points to identify your target compound. Mass is very stable, but may not give you all the information you need. For instance, many chemicals have structural isomers that share the same mass, but can have drastically different effects on the human body. Retention time, on the other hand, is not very stable. Retention time will change whenever you change experimental conditions, but even when you're using exactly the same conditions, retention time can still drift between batches or within the same sample batch. Thus, when performing targeted experiments, it's essential to run a purified standard alongside your samples every time you perform an analysis to ensure that you are identifying the correct compound. But this presents a problem. If you're only looking for compounds that you've already identified and purified, you're never going to identify new contaminants. This is where MSMS can be incredibly helpful. Unlike targeted analyses that only perform MS1, untargeted MSMS analyses perform an additional round of mass spectrometry that breaks apart each molecule and measures the mass of the fragments it produces. This creates a chemical barcode that can be used to identify both known and unknown compounds. Here's an example of how this works. Today we're analyzing 2 batches of salmon, one wild-caught and one aquacultured, to find out how they're different and how those differences might affect a final product. The first thing we'll do is run molecular networking, which is our all-in-one workflow that allows you to identify, annotate, and network your mass spectrometry data. All we need to start our experiment is our MSMS data, which I'm going to add in 2 groups like so, plus the blanks. You'll see there are a lot of other parameters here, which you can read more about in our documentation, or you can go to our Running Your First Molecular Network tutorial video. To start the analysis, I'm simply going to click Submit Workflow here. So that analysis took about 12 minutes to run and identified over 3,000 metabolites in these samples. About 600 of those have library annotations, which means that the MS2 barcode matched to the barcode of a known compound. However, we do have a way to glean information about all those unknown molecules using molecular networking. Molecular networking groups structure-related compounds. You can open up the molecular network using the Visualize Full Network in Browser button here. In a network, each metabolite is represented as a circle or node, and compounds that have structural similarity are connected by an edge between the nodes. Blue nodes have library matches, while gray nodes don't. However, by clicking on the edge between a blue and gray node, we can compare those MS2 spectra and assess their similarity. For example, you could find the node for your target compound and check out if it has any connected nodes, which are likely to be degradation products or other structure-related compounds. One thing to note here, if you're an experienced user, is that we use the MS2Deep score algorithm to make this network, so spectral similarity may look a little different than the same network using our standard cosine scoring. While networking can identify both your target compound and any novel contaminants, statistical analysis can provide information on how the relative abundance of those compounds changes over time. You can run statistical analysis directly from the network by clicking on the button here. This will auto-populate all the correct files, and all you have to do is enter in these options here and click Submit Workflow. You can always use the links below to pull up the data and check it out yourself, but for now, let's look at the volcano plot. To open up the volcano plot, click the button here, and then select which groups you want to compare. This shows us all the metabolites that are differentially abundant in wild-caught and aquacultured salmon. If you want to find a specific compound, you can search for it here. If you click on the compound, you can highlight that compound in the plot and draw a box plot comparing the different groups. Now, finding and measuring differences between compound abundances is great, but how do you figure out where these compounds came from? This is especially important if you've identified a contaminant and want to know what batch or source material it originates from. For this, you can use the Mass Spectrometry Search Tool, or MAST. For today, we're going to do a mass search for spermine, but you can use MAST to search any MS2 spectra, whether it has a library match or not. There are a couple of ways to do this in Ometa Flow, but for today we're going to use the standalone workflow right here. All you need for this is whatever MS2 spectra you want to search in MGF format. You can also choose how tolerant you want your search parameters to be and whether to search for analogs of your target compound. So this search took about 45 seconds to return over 1,000 matches to our spectrum. Now, our server is set up to search the public repositories in GNPS, which can give us information on what types of sample, species, or body part a molecule has been found in. However, it is possible to pair MAST with custom databases to return information on where a compound was detected across plants, source material, or time. If you have more questions about custom MAST databases, please contact us using the link below. So that's our overview of how you can perform unbiased QC analyses using MSMS data and molecular networking. Feel free to check out the results of each of these workflows using the links below. Thanks for watching.